In the previous lecture, we have learned about the difference between chemical and electrical transmission, and we have learned about the basic steps that occur during the chemical transmission. In this second lecture, or in the next series of the lectures, we are going to go in a little bit more detail through each of the steps, or many of the steps that are involved in the chemical transmission. In particular, during today's lecture, we are going to look at a neurotransmitter synthesis, packaging, and release. And the goal of this one will be to understand how use of different neurotransmitter diversifies chemical transition, to learn about two major categories of neurotransmitters, how they're synthesized, how they're packed into the vesicles, how those vesicles are delivered to the active zones, and also we will introduce the molecular network, the protein network that's present at the active zone that plays a role in the docking, priming, and also the release of neurotransmitter during when action potential arrives. Now, chemical transmission is defined by the release of the chemical called neurotransmitter into the synaptic cleft and then transmit information from presynaptic to postsynaptic neurons. And what's really crucial in this way of interaction between neurons is that having more than one neurotransmitter significantly diversifies the repertoire of chemical synapses, what they can do indeed. First, different neurotransmitter can produce different types of responses. On the individual neurons, for example, certain neurotransmitters such as glutamate is going to be excitatory. On the other hand, we can have different neurotransmitters such as GABA, which will be inhibitory on the same neurons. So release of different neurotransmitters will have different types of responses. Another very interesting characteristic is that speed of postsynaptic responses produced by different neurotransmitters actually can differ. For example, different neurotransmitters, in particular, let's say the smaller ones such as glutamate and GABA, usually mediate those rapid synaptic changes. On the other hand, neuropeptides usually mediate much slower synaptic responses. So therefore, you can have different times, you can control synaptic activity over different times. Finally, until very recently, it has been taught that neurons can release and synthesize only one given neurotransmitter. Now we know that many neurons actually can synthesize, produce, and also release two or more different neurotransmitters at the same synapse. And when this happens, it's called co-transmission. Now, because different neurotransmitters do not need to be packed in the same vesicles, they actually need not to be released at the same time or simultaneously. Actually, the release rate can depend on the rate of the activity of the neurons. One example are going to be, again, small molecules such as neurotransmitters such as glutamate, which are packed in the clear vesicles, and they're usually released. Even the low rate of activity is going to lead to release. On the other hand, neuropeptides usually need higher rate of activity to be released. Therefore, depending on the rate of release, you may have different chemical signature of these same synapses. Now, over the time, there have been different criteria that have been used or developed to define what different chemicals can be called neurotransmitters. And here, example of 3 different ones that we are going to be discussing a little bit more. Number 1 criterion is that that chemical need to be present within the presynaptic neuron. This is kind of obvious. If a presynaptic neuron is going to release something, it needs to be made or it needs to be present in the presynaptic neuron. This can be usually done by immunolabeling, either with immunofluorescence or immunogold labeling, also in situ hybridization. Additional thing that help with this is to show that the enzymes, biosynthetic enzymes that are required to make that neurotransmitter and also precursors are present. That is considered to be additional evidence for that. One exemption for this rule are going to be glutamate, aspartate, and glycine, because they are amino acids which are also involved in other metabolic reactions inside of the cell, and therefore just having them present in neurons do not define that they will be released by that neuron. Second criteria is a little bit harder to achieve, to show. Second criteria says that chemical needs to be released in response to presynaptic depolarization, and that release has to be in a calcium-dependent manner. One of the reasons why is this a little bit more technically challenging to prove is that most of the neurotransmitters after their release are quickly removed from synaptic cleft, either by diffusion, the reuptake, or the enzymes. And the third criteria is that receptors which react to that neurotransmitter need to be present in the postsynaptic cells. There are different ways which one can show that. One can be application of the neurotransmitter. Now levels of neurotransmitter can be applied, and then you can see whether you get the same response as you get during chemical transmission. You can also use additional evidence. We'll be using agonists and antagonists either to induce or to block the activity of certain neurotransmitters. Now, using this kind of criteria, there have been over 100 chemicals that have been identified to be neurotransmitters or putative neurotransmitters, and they can all be broadly divided into two big categories of substances. They can be divided into small molecule neurotransmitters and neuroactive peptides. It's really mostly based on their size. And in this table, we have example of small molecule neurotransmitters and also peptide neurotransmitters. Actually, in both of these, and the second table will show you some major functional features. If we look at the table on the left, we have the examples of small molecule neurotransmitters, such as that can be divided in a few groups: acetylcholine, then amino acids such as glutamate, aspartate, GABA, and glycine. Then we have biogenic amines and catecholamines, which have dopamine, norepinephrine. Epinephrine is one big group defined by the catechol ring, which is the benzene dihydroxybenzene. Then we have serotonin, histamine, and we also have purines such as ATP and its derivatives. On the bottom is the example, just a small example, peptide neurotransmitter, which are defined by having usually 30 to 36 amino acids long. And you can appreciate the difference in size between small molecule neurotransmitter. We have these spatial Phelan models for glycine and norepinephrine, and then small methionine caffeine, which has 5 amino acids. You can see that they are much bigger. In the table next on the right, you have different neurotransmitter, example of different neurotransmitter and different postsynaptic effect they have, with the cautionary note that these are the most common postsynaptic effect. Actually, the same neurotransmitter can elicit different responses, but this is the most common one that's present here. What is also interesting in this table, you have the information about the precursor and the rate limiting step in the synthesis, and also removal and what kind of vesicle they are going to be packed in. Now, we are going to go discuss this in a little bit more details very soon. Now, for chemical transmission to be really efficient, the amount of neurotransmitter release into synaptic cleft has to be really closely controlled, and therefore neurons have developed a really sophisticated means by which they will control the amount of release by controlling the synthesis of neurotransmitter, packaging of neurotransmitter into the vesicles, and then delivery of those vesicles at the active zones, and obviously their calcium-triggered release. In addition to the release, the control also the reuptake or the levels of neurotransmitter after its release is controlled through 3 different mechanisms, either through reuptake, diffusion, or degradation. So all these 5 different steps, or we can divide it more, really constitute the life cycle of neurotransmitters. So it has first to be made, it has to be synthesized, and it has to be packaged into the vesicles, delivered to the active zone, which then those vesicles will fuse when calcium arrives. And transmitter is going to bind receptors, but the leftover has to be either reuptaken or diffused or enzymatically degraded. Another thing that undergoes recycling are synaptic vesicles, shown here for the smaller synaptic vesicles. And again, we are going to be talking in more details about each of these steps. So we are going to start with the synthesis of catecholamines, which are dopamine, norepinephrine, and epinephrine. They're all made from the tyrosine, and there are 4 major enzymes that are involved in the synthesis, depending which of these neurotransmitters the neuron is going to be using. In the first step, tyrosine we are going to add 1-hydroxy group using tyrosine hydroxylase enzyme, which also is using biopterin. This enzyme is a rate-limiting step in the synthesis of all catecholamines, and this is when we say rate-limiting step, it's the enzyme that's really regulatable, whose activity is regulated by levels of activity. For example, when dopaminergic neurons are activated, when they increase their activity, they are first going to phosphorylate tyrosine hydroxylase. So it's going to be post-descriptional modification, which is going to increase its activity and therefore produce more dopamine down the line. If they are actually activated for a longer time, for example, under extreme stress, then these neurons are also going to increase the synthesis of messenger RNA for tyrosine hydroxylase and therefore increase the number of the proteins or number of the enzymes that can catalyze this reaction. It's important to know that biopterin is really required for this reaction, and if you have problems with biopterin, you will also have obviously the problems in making dopamine. So this first reaction, hydroxylation of tyrosine, is going to lead to formation of dihydroxyphenylalanine or DOPA. The next step in reaction, next enzyme is DOPA decarboxylase, which removes a carboxyl group to make dopamine. So in the neurons that make dopamine, you only need those two enzymes, and only those two enzymes are going to be expressed, tyrosine hydroxylase and DOPA decarboxylase. On the other hand, in norepinephrinergic neurons, you're going to have additional one enzyme called dopamine beta hydroxylase, which is going to remove, it's going to add one hydroxy group to dopamine to make norepinephrine or noradrenaline, which is really one very interesting characteristic for dopamine beta hydroxylase. Is a membrane-associated enzyme, and it's actually the only membrane-associated enzyme in this whole neurotransmitter synthesis. But what this means is that norepinephrine is the only neurotransmitter synthesized within vesicles. So dopamine goes into the vesicles where norepinephrine is made. And then in a few central nervous system neurons which make epinephrine or adrenaline, they require one more step and one more enzyme called phenylethylamine and methyltransferase, which is adding one metal group to norepinephrine to make epinephrine. And again, whether cells will produce dopamine, norepinephrine, epinephrine is really determined by the expression of these enzymes. As I said, tyrosine hydroxylase and DOPA decarboxylase are required and present in neurons that produce dopamine. If they're going to produce norepinephrine, they need dopamine beta hydroxylase as well. And then if they're going to produce epinephrine, they also need phenylethylamine and methyltransferase. Serotonin is made out of essential amino acid tryptophan in a very similar steps that are required to make dopamine. There is tryptophan hydroxylase, which also using biopterin requires it. And then the next step is going to be, again, 5-hydroxytryptophan decarboxylase. Those decarboxylases are otherwise known as aromatic L-amino acid decarboxylase. So it's a very similar step of reaction that is involved in making serotonin as dopamine, and those enzymes are very similar. Acetylcholine requires only one enzymatic reaction in which enzyme called acetylcholine acetyltransferase uses choline and acetyl coenzyme A, which is one of common metabolites, to make acetylcholine. What is interesting that neurons cannot synthesize choline, so it has to be derived either from the diet or it has to be recycled. And almost 50% of the choline is derived from either food or from acetylcholine hydrolysis. And the hydrolysis that we're talking about is that after acetylcholine is released from synaptic vesicles in the synaptic cleft, there is additional enzyme called acetylcholinesterase, which is going to then cut it acetylcholine acetate and choline can be reuptaken by the neuron. So 50% of choline comes from the food and 50% comes from this reuptake after it's released. And then we have synthesis of glutamate and GABA. Now, this is a very interesting step because these are amino acid transporters, or glutamate is amino acid transporter, and glycine as well, and aspartate, so they're present in all neurons, but they actually act as neurotransmitter only in some of them, and therefore the both metabolic and neurotransmitter parts of glutamate have to be separated in the cell. Most of the glutamate in the neurons is going to be made from alpha-ketoglutarate by enzyme glutamate synthase. Alpha-ketoglutarate is part of the citric acid cycle. It also can be derived from the glutamine that's imported from the glia. By enzyme glutamine synthase is going to then remove the amine group and make the glutamate. On the other hand, for GABA urge, GABA is not present in cells, it's just present in cells that secrete GABA, and it's usually made from glutamate, and it requires additional enzyme called glutamate decarboxylase to make GABA. And there are at least two known, GAD65 and glutamate decarboxylase 67. So those enzymes are required, and they also use vitamin B6 as a cofactor. The importance of vitamin B6 in the reaction of making GABA has unfortunately been implicated very well. It has been shown very well in this disastrous series of infant deaths that was caused by omission of vitamin B6 into the infant formula, which then led to decrease in the synthesis of GABA, which then led to the decrease in the GABA release in the brain and led to the seizure in these infants, which resulted in quite a lot of deaths. And finally, we're going to briefly discuss the processing of neuropeptides. There are several different neuropeptides that can be encoded by a single messenger RNA, which is then made into pre-propeptide. This pre-propeptide is going to be having the signal peptide that's going to define it as a secretory molecule in the endoplasmatic reticulum. It's going to be transported to Golgi and then packed into vesicles. And then inside of the vesicles, there are going to be different enzyme peptidases, which are going to cut it into smaller pieces. Now, there are two different major reasons why this can happen, or two different theories. Polypeptides can generate actually different distinct peptides, so we can have several different peptides made from one, which are going to release from the same vesicle to have a really complex response of postsynaptic neuron. In addition to this complexity, you may lack some peptidases. So, for example, if you start for propiomylocortin, present here in A, you may end by ACTH or beta-lipotropin. But then if you have additional peptidase, or if the cutting sites are free, you may end up with gamma-lipotropin and beta-endorphin secreted from these vesicles. Another one is diversity of responses and complexity of responses. The other reasoning why we think this happened is that you actually can amplify. So as you can see, for pre-proenkephaline, it actually can mean 6 different copies of metenkephaline from one propeptide made. So you can increase the amount released. Now, the synthesis of small molecule transmitters and peptides differ in several different ways. One is this place where it's made, so they're spatially different. So for example, for small molecule transmitters, which you see in the top panel, enzymes that are involved in the synthesis, for example as GAD65, are made in the cell body, but then they are transported by slow axonal transport along the microtubules to the nerve terminals, which is where the enzymatic reaction that make GABA from the precursor occurs. And then they are packed usually in these small clear core vesicles that contain these small molecule transmitters. When they are released, then the synaptic vesicles are recycled, and then neurotransmitter can be again repackaged. On the other hand, peptide transmitters are usually synthesized, as I said, in the cell body, and they are synthesized in the endoplasmatic reticulum. Then they move from Golgi and are transmitted in the vesicles. These vesicles bud from the Golgi apparatus, and they are then transported to nerve terminals along a microtubule using the fast axonal transport. Once they come, they are actually released and they cannot be recycled. So, and the neurotransmitter, peptide neurotransmitter, are also not reuptaken. When they release, they diffuse. They're also packed in a differently looking vesicles. Instead of small clear cold vesicles that we have for the small neurotransmitters, we have these dense core vesicles. And you can also see that they are not present exactly at the active zone. They're present a little bit further away from the active zone, which also defines how they are released, which we are going to be talking in next lecture. So the next step we're going to go through is the packaging of neurotransmitter in the synaptic vesicles. Now, neurotransmitters, when they're made, they need to be transported into the vesicles, and they need to be actually concentrated in the vesicles, which require transport against their electrochemical gradient. And this requires transport proteins. So what occurs in the synaptic vesicles, we have vesicular transporters that are really specific for each of the, or for the groups of neurotransmitters, small molecule neurotransporters. And we also have a proton pump which fuels neurotransmitter uptake because it generates a protein electrochemical gradient. It uses energy of ATP, hydrolysis of ATP, to the movement of protons into the vesicles, thus making the also pH of the vesicles much more acidic than the cytoplasm. Now, the final concentration of neurotransmitter and how much, of course, it's going to be released by single synaptic vesicles, which is going to define how much effect it's going to have on postsynaptic neuron, is going to depend on cytoplasmic concentration of neurotransmitter, so how much of neurotransmitter is made, which is going to be defined by the amount of precursor present, by also the activity of the enzyme. It's going to depend on activity of the transporter itself, so these transporters' activity is also regulatable. It's going to depend on the magnitude of the driving force, which we said is also made by these proton pumps, and in their activities, because it requires the energy of ATP, will be depending on how much energetic status of the neuron. And finally, it's going to depend on non-specific leakage across the vesicle membrane. So there are 4 major groups of transporters, as I said. For each group, so we have VMATs, which are for monoamines, so they secrete those transporters. There are 2 of them. Number 2 is present in neuronal cells. They are important for transport of dopamine, norepinephrine, serotonin inside of the vesicles in the CNS. We have VACHT, which is required for transport of acetylcholine inside the vesicle. VGAT transport both GABA and glycine, both inhibitory small molecule neurotransmitters inside the vesicles. And then we have VGLUTs. We have 3 different, and they are actually expressing different neurons, which are in transport glutamate inside of the vesicle. There's one very interesting characteristic about this transport, because we said it depends on the electrochemical gradient of protons, which can be divided into chemical and electrical gradients. And depending on which neurotransmitter they are transporting, these transporters really vary in their dependence on the chemical or electrical components of this electrochemical gradient, as is shown in this slide. So let's look, for example, in A. We have VMATs. Again, they are transporting monoamines. So monoamines are positively charged. So it's a co-transport, so transport of one monoamine, which is positively charged molecule, will result in leakage or release or leaving of 2 protons from the vesicle, so that's the cost. So that leaves us in change in electrical gradient just of 1, because 2 positively charged molecules are leaving, 1 is entering, so we have a smaller change, but 2 in the pH, in the chemical gradient, so we actually have 2 protons leaving. Therefore, these VMATs actually, their activity depends more on the pH than it depends on the electrical component of electrochemical gradient. For the neutral neurotransmitters, such as VGATs, they equally depend on both chemical and electrical components. On the other hand, for glutamate, which is negatively charged, here the situation is different. When glutamate enters, and let's say it's really not known how many protons are required to leave for one glutamate. That's why in this T is present as N of H. We just don't know. But let's suppose it's N is equal to. So let's suppose we have 2 protons leaving for one negatively charged glutamate to enter. So the change in the charge is here now -3, because we have 2 positively charged protons leaving and 1 negatively charged glutamate entering. So we have big change in the charge. But as for the pH, we have 2 protons leaving. So therefore, this activity depends, we have a bigger change in the electric component than the chemical component of the electrochemical gradient. And therefore, VGLUT2 activity or transport of glutamate depends much more on the electrical gradient. This plays a big importance because you can then regulate the activity of these transporters, and by regulating their activity, you can regulate the amount of neurotransmitter that's going to be packaged into each vesicle, by changing each of these components. So for example, if you change electrical components, then you're going to make the proton pump works much more and acidify this more. So what's VMAT vesicles that transport monoamines, they can use chloride channels, so they can import chloride to change the electrical component and then make the pump works much harder and therefore increase delta pH. They can also transport glutamate inside, so if the dopamine containing vesicles are going to express some V-glut, a transporter that are going to allow some glutamate to come inside, it again is going to increase their pH and therefore allow more dopamine to be packaged into each vesicle, so therefore increase the amount of dopamine that's going to be released. The next step in the cycle is vesicle docking and priming for release, and after we as we transport neurotransmitter inside of the vesicle and concentrate it significantly, now we need to deliver these vesicles at the active zones, and active zones, as we know, are those electron dense material on this electron micrograph, and which release of neurotransmitter happens. This is the place where neurotransmitters are released into synaptic cleft upon calcium entrance or upon arrival of action potential. So we're just going to discuss a little bit the protein network, the way we reached or increase our knowledge or understanding of what happens at the exocytosis of synaptic vesicles actually started from biochemical work that defined many of the protein that play a role in this. Proteins were isolated from either active zone or from the synaptic vesicles. So in this next slide, we are just going to look at the proteins that are found in synaptic vesicles, just some of them that play a role in exocytosis. We have here neurotransmitter receptor. If we start from right to left, neurotransmitter transporters that we just talked about, that again are involved in the transport of the neurotransmitter inside of the vesicles. Then we have a protein called synaptobremin, which is important for the fusion. So it's in this box we have SNER complex. SNER complex is made out of 3 proteins. One of them, synaptobremin, is present at the vesicle, and the other 2, SNAP25 and Citexin, are present at the plasma membrane. SNER complex is made out of those SNER domains, which are the rectangles present in these proteins, which interact with each other to mediate, they're required to mediate fusion. So SNER complex is an extremely important complex in the fusion. Synaptotagmin, right next to it, is thought to be the calcium sensor for exocytosis. We're going to talk more about it. And then we have Rab proteins on the synaptic vesicles, which are thought to be important into vesicle docking and priming. They interact with the components of the active zone that we're going to talk in a second about, RIM protein and MUNC13. And then we have synapsins, which have been shown by Paul Greengard that they actually play a role in recruiting the vesicles from the recruiting vesicles to be delivered at the active zone. So what is the active zones protein that play a role? This again very complicated diagram image in which we have also synaptic vesicles and the active zone proteins. I want you to pay attention to the RIM proteins. RIM proteins are the pink ones, which form this another complex. So we talked about SNER complex, which is really important into the fusion and exocytosis, but the RIM complex seems to be very important in the vesicle docking and priming. RIM protein interacts with many other proteins in the active zone. It interacts with the Rab proteins on the synaptic vesicles, and that's how it's thought to dock the vesicles, but also interacts with MUNC13, which is involved in the fusion, and also RIM binding proteins and RIM protein, both of them interact with the calcium channels, and they are thought to actually tether calcium channels very close to the vesicle docking site. Therefore, the calcium that enters through these calcium channels is going to be right there next to synaptotagmin to trigger the fusion. Other molecules at the active zone that we have some knowledge might be playing a role are PICOL and BASOON. They're really big proteins that are thought to guide synaptic vesicles to the active zone. Then we have proteins, which are the ALKs and aliprins, which are thought to contribute to the precise localization of active zone across from the postsynaptic specializations. But it's really a challenge is to fit this in a spatial, how this really looks in the active zones. And what we have here actually really amazing images of wrong neuromuscular junction using electron microscopic tomography. In the top you can see vesicles, and then you have these structures which were called beams and ribs that are binding to them. And then also we have pegs, which we think are calcium channels. Now what is really a challenge right now is find the molecular identity of these structures that we can see through electron microscopy tomography. It is quite possible that Riem and Monk protein, which we have talked a moment ago, are involved in docking the vesicles, and also calcium channel are molecular, or at least some play part of molecular components of beams and ribs that we can see in this neuromuscular junction. Now we'll talk about vesicle priming. So after the vesicles are docked by interactions of Riem protein and Reb proteins at the vesicle, then we have the priming. What priming means is these vesicles are ready to be released when calcium enters, so they're really at the right place, and there's just very small amount of event that has to happen for the release of neurotransmitter to occur. But vesicle priming, what we think vesicle priming means, again, we do not have a full understanding of these events. I think that means a formation of the snare complex between, as I said, synaptobrevin, snare domain of synaptobrevin, and SNAP25 and Cetaxin, which are associated or present in the plasma membrane. The current idea or current model saying that this snare complex formation brings plasma membrane of synaptic vesicles very close a position with the presynaptic plasma membrane, then bring them so close that it will allow fusion whenever the calcium enters. And you have also here presented synaptotagmin with the calcium binding there. So therefore, molecular mechanism of exocytosis here is present in these images. So the idea is, again, that vesicle docks, probably through interaction of Riem proteins and Rebs on the vesicles. And when vesicle docks, the priming happens. So we have interaction between synaptobrevin, Cetaxin, and SNAP25, making of snare complex, and then pull these membranes very close together. Then the calcium enters, it binds synaptotagmin. Synaptotagmin has two C2 binding domains which bind calcium, and then binding of the calcium to synaptotagmin also increases its binding to phospholipids. So it's thought that these conformational changes triggered by calcium binding to synaptotagmin are going to then trigger membrane fusion or opening of the membrane pore that's then going to be opening further. After fusion, of course, this complex needs to be dissociated so that we can have this reuptake and recycling of the vesicles. For this, we require two protein complexes or another protein complex, NSF and SNAP proteins. These are different than SNAP25 proteins. The name is just very similar. And NSF and SNAP also use energy of ATP to dissociate snare complex, so they can be reused in the next cycle of vesicle fusion. So what I want you to think about this is a problem actually about how the neurotransmitter synthesis is really important, what kind of symptoms it can lead. So this problem, I'm introducing Stygarda syndrome. This is a rare genetic disorder which is characterized by abnormal gait and also involuntary muscle contractions. It's caused by a loss of function mutation gene GSH1, which encodes enzyme which is required for biopterin synthesis. I just want to put the synthesis of both dopamine and serotonin here to show you the step in which tyrosine hydroxylase and also tryptophan hydroxylase require biopterin for the enzymatic reactions. So based on this, I want you to think, would you expect any changes in catecholamine release in patients suffering from Stygarda syndrome? And how do you think can lead these abnormal gait and involuntary muscle contractions? And what about serotonin? And then how would you treat Sagawa syndrome? What, based on these, again, schematics, what do you think would be the proper treatment for